All right. Okay. Good morning or good afternoon, everyone. It's a pleasure to participate this winter school. And today I will give you an overview of what we are doing here in Taiwan about knotty proteins. So our lab is very interested in a family of topologically knotty proteins. And I will explain to you in the next hour or so how we define these proteins as being topologically knotted. At the same time, because of COVID, we also spend a lot of time to look at another family of knotty proteins. They are not literally knotted, but they are really a big issue. So our lab in the past few years have been working on the structural characterizations of the spike protein. That's the protein on the surface of coronaviruses, and how they interact with host receptors and monoclonal antibodies as a way to develop antivirals. But in the spirit of SKCM², today I will focus my talk on the topologically knotty proteins. I suppose many of you are not familiar with the biological backgrounds of protein structures. So I will spend some time in the beginning of my talk to give you an overview of the basics of protein structures and how we study the physical chemical properties of proteins. So to outline today, I will give you in basics the ABCs of protein folding and topology, and define topologically knotted proteins in the context of the structural biology. And I will give you an overview of how we study protein folding and how they knot. And in the second half of my talk, I will give you some flavor of the biological significance and the protein engineering side of protein knots. So proteins are the very essential elements of our daily life. We have all kinds of proteins that carry out different enzymatic functions or transmit signals from the outside to inside or inside to outside. So here is a collection of experimentally determined protein structures that are deposited in the protein data bank, known as the PDB. So you can see that we have some very large viral particles that look like spheres. We also have the muscle cells that are very elongated. Protein binds to DNA or embedded in the membrane. Now, the first protein structures was solved by X-ray crystallography by Max Perutz and John Kendrew at Cambridge University in the early 50s. And because of their groundbreaking discovery, they were awarded the Nobel Prize in Chemistry in 1962. So here on the right, you will see the atomic model they built for the first ever protein structures that is the protein that carries the oxygen and is purified from the blood of sperm whales. It's a moderately sized protein of a few hundred to 1,000 atoms or so. But to solve this protein structure requires the ability to crystallize individual proteins into a lattice structure and then to get diffraction patterns to solve these atomic structures. These days, we have, in addition to protein crystallography, NMR spectroscopy that can be used to study protein structures and dynamics in solution, and that led to the Nobel Prize in Chemistry in 1991 and 2002. And the very recent breakthrough in structural biology is the use of electron microscopy to look at very large protein assemblies. And because of the importance, the three scientists were awarded Nobel Prize in Chemistry in 2017. I will not go into the details of these technologies, and if you're interested, please go to the Nobel Prize websites for some sort of introductory material for reading. Fortunately, in Academia Sinica here in Taiwan, that is located in Taipei, north of Taiwan, we have easy access to X-ray crystallography, which is in Hsinchu, that's within an hour car drive from Academia Sinica, or high-field NMR spectroscopy and cryo-EM facilities on our campus. So if you're interested in using these infrastructures, we are very happy to host you to establish collaborations to look at your system of interest. In fact, through the SKCM² initiative, we already established some collaborations with other PIs within this SKCM² to look at their system of interest, not only limited to proteins, we also look at supramolecular assemblies that are not based on biological systems. So coming back to the evolution or the development of structural biology, as I said, the first crystal structure of protein was determined in the mid-50s, and then the first NMR structure was reported in the 80s. The development of cryo-EM or EM in general was relatively late, but it is now undergoing a explosive growth, as you can see in this dark orange line, that the number of cryo-EM structures reported every year is now exceeding the NMR structure and is approaching and potentially overtaking the output of X-ray crystallography. It is important to note that in the early days, it is actually very time-consuming and costly to determine protein structures. So when the Human Genome Project was completed in the early 2000s, there was a global call for structural genomics consortia to try to solve as many protein structures as possible as a way to understand how our genome functions. So the idea is that if you know how the individual building blocks of our body looks like and work and interact with each other, we will be able to understand how our body functions in greater detail. But to invest in the structural genomics consortium, on average, it would cost over $150,000 per structure. So in fact, nowadays we have over 150,000 protein structures deposited in the protein data bank. So this is really a gold mine that is waiting to be explored by all kinds of scientific disciplines. Well, you probably have heard about this program called AlphaFold that was developed by DeepMind, funded by company Alpha. So AlphaFold was released in 2022, and in that year they provided over 200 million predicted protein structures to the public. And this is an enormous breakthrough in terms of structural biology, because these days we are no longer limited by the experimental resources or the manpower to solve all the structures. You can actually predict protein structures, and the software will tell you whether it is confident in the prediction, or in some cases, I will see here, if you type in a name, for example, BAP1 is one of our target protein, you will see that the core region, you have helical structures that are colored in dark blue, meaning that AlphaFold is very confident in the structure prediction in that region. But there are also regions that are colored in yellow or even orange, and that means that the confidence score is below 70 or even below 50. So users need to be mindful when they use the predictions generated by AlphaFold to see which part is of high confidence and which part is of low confidence. Regardless of these uncertainties, AlphaFold presented a huge database of protein structures that are available to the public to look at the structures predicted by AlphaFold, and you can go to this database to check your protein of interest, whether it's there, and how does it look like if it's not yet been experimentally verified. So I will spend some time to go to the ABCs, or the basic alphabets of protein structures. So protein structures can be categorized in 4 different levels, from primary, secondary, tertiary to quaternary structures. Basically, the DNA in our body or in any genome encodes the 4-letter ATGC sequence that will be translated into 20 different kinds of amino acid as a linear chain. This is called the primary structure. The combinations of these polypeptide chain can fold into helical structures, or as I highlighted in yellow on the upper right corner, beta strand-like structures. These are called the secondary structure. So you can connect these secondary structure sequentially, and that will fold into a tertiary structure. So if it's a complex formed by more than 2 proteins, which I color magenta and green on the lower left corner, that is called a quaternary structure. So in this case, it's a binary complex of a green protein binding to the magenta protein. So that's the common definition of protein structure at 4 different levels. We have the alpha helices. They are formed by a series of hydrogen bonds in the backbone amide to the carbonyl. I highlighted in dashed line on the right. You can also have multiple strands of beta strands to form a sheet-like structure. Between the strands, there are also hydrogen bonds, and then you can see these dotted lines to connect the hydrogen donor and the acceptors. So that's a different kind of secondary structure. So a beta sheet will contain at least 2 beta strands. And in this example, we will have 3 beta strands to form a sheet-like structure. So a linear polypeptide chain can sample different kinds of torsion angles that are defined as the phi-psi angles. So the 2D plots of the phi-psi combination, you will see that there are actually a limited space that are sampled by polypeptide chains. And you have the alpha helical region that has a different type of combination compared to the beta sheet-like structures. So coming back to topology, so this is quite different from the topology we, or some of you are using in mathematical terms or in topological material. So in protein structure, a topology is defined as the way the individual secondary structures are connected in space. So what you will see here is that I have a number of helical structure symbolized as these cylindrical red cylinders. I also have individual beta strand colored in green. So from the beginning, that's the indicated as the N, you will go through a series of sequential arrangements to the C, that's the end of the polypeptide chain. So depending on the way these secondary structure elements are linked, you will have different types of topologies. So here is another example. Antibody have these common Ig domains. They are primarily formed by a series of beta strands. And in nature, there's many kinds of beta-stranded proteins that are connected in different ways. So in a way, if you look at these beta strands, they are in fact defined as having different topologies. So if we have a protein that has 100 amino acids, each amino acid can sample 3 phi angle and 3 psi angle. So for this 100 amino acids, there will be a combination of 3 to the power of 198 combination. That's approximately 3 to the power of 94. This is a huge number, meaning that the degree of freedom of a not very big protein, 100 amino acid, is vast. So if we were to sample all the conformation using a supercomputer that would be able to calculate 1 petabit per second or 1 petaconformers per second, this will still take longer than the age of the universe to sample all the possible conformation. So this is really not a realistic random sampling process. So this is the idea is coined by Levinthal in 1969. So what he said is that protein folding is not a random process by random sampling. There must be some information encoded within the protein sequence to guide the folding process in an efficient way. So on the left, that's the same crystal structure I show you. That's the first protein crystal structure. And on the right, that's Max Perutz in his later days, taking exactly the same chemical composition of this protein as a linear chain. So the challenge is whether we can efficiently and correctly fold this long polypeptide chain into a defined structure. And this is known as the protein folding problem. So the second protein structure that was solved is lysozyme. That's the primary constituent in egg white. And David Phillips in the UK, he also gave the audience there an idea that on his right, that's the folded lysozyme crystal structure. On his left, this the same polypeptide chain hung from the ceiling of the auditorium. You will see that this linear polypeptide chain will need to undergo a series of folding events to fold into this very compact structure that is solved by X-ray crystallography on his right-hand side. So how do we show that proteins can fold efficiently and spontaneously? And that was actually a simple experiment Christian Effinson did in the 60s. So what he did was taking a protein called RNase. This is an enzyme that would digest RNA into small pieces. And he added reducing agent, beta-mercaptoethanol, and urea, which is a chemical that would unfold protein structure in the test tube. So the protein becomes unfolded, and therefore it cannot execute its enzyme activity. He then removed, again, in the test tube, the reducing agent and the chemical denaturant, and then this RNase can execute its enzyme activity again, meaning that this polypeptide chain is able to fold into the native structure spontaneously without additional factors. In other words, the information of how to fold the structure in a precise way is encoded within the primary structure of this protein. So if we think about the concept, we can think about in free energy landscape terms. To fold properly, we can imagine it is being in a low energy stable conformation. So rather than throwing a dice to see how the energy look like, is it stable or not, the people have come up with this folding free energy landscape concept. So you will have this funnel-like structure. So you will start potentially in high energy conformation, but because the free energy landscape is a funnel-like, so protein would then slide into the low energy native conformation. So on the upper left corner, that's the ideal scenario. This is a very smooth funnel. So it does not matter where you start, you will end up with the native conformation. But in real life, protein folding free energy landscape is not very smooth. So sometimes they are rugged. There are a lot of local minima. And in some extreme cases, we have examples that can sample distinct conformations that are sort of bistable. And that's exemplified on the lower right corner. You could have multiple conformations that are comparably stable, and to jump from one conformation to the other conformation, you will have to overcome a large energy barrier to sample the other conformation. So this becomes the concept of this free folding funnel that was coined by Wallace, Onuchik, and Thermalai. And later, with the help of powerful computers, Martin Koplas, Christopson, and Michele Valtruscolo then studied the folding events of different proteins in atomic details. And indeed, what they saw was that you can start from a random coil, meaning that there are not defined structures in the high energy state, but very quickly they will collapse into a transition state and then fine-tune its structure to fall into a well-defined native structure. All right, so that's a general overview about protein structure and protein folding. So the question here is, can we or can you tie a protein knot? So as we just learned, proteins are long polypeptide chains. And I think this is our common daily experience. If we have long strings in a confined space, upon agitation, we are bound to have some entanglement. So this was actually tested by physicists and reported in this PNAS paper. They showed that long strings would have certain probabilities to tie knots spontaneously upon agitation. And the same question or the same observation was found in the 70s when structural biologists were looking at different protein structures, try to classify them in the context of topology. So in 1977, the first knotty protein was identified by crystallographers, and they found that there is a 3-1 knotted conformation. I would come back later to tell you how that knot is defined. This is slightly different from the mathematical way of defining a knot. So it's actually quite early days, almost 50 years ago, people realized there's a knotted protein structure in nature. But most people are still very skeptical about the identity of these protein knots. So in 1994, Mark Mansfield then submitted a letter to Nature Structural Biology. And he started by saying that most biochemists would probably agree that proteins in native states are not knotted. This sounds a bit paradoxical. The reason is that it is inconceivable for protein to undergo this repetitive snake-like motion to basically to go along its backbone, find a loop and thread through it and tie it, as I schematically shown here on the right. Nevertheless, he tried to develop an algorithm to survey all the protein structures that were available in the database at the time. And in the early 90s, there were only about 400 protein structures available. So what he did was try to simplify or smooth the protein backbone paths. So if it's a helix, you can stretch it into a straight line, but the only condition is that you cannot cross or you cannot jump from one path to the other. If it can no longer be stretched into a straight line, that would be considered as being knotted, as exemplified in the lower right corner here. What he found was the same structure people reported in 1977. That is a three-band knotted structure. So these days, we have different tools to help us visualize whether a protein is knotted or not. And on the upper panel, that it is a 3-1 protein knot. So as I show, I hope you can see here. So if this is a helix, we can simplify into a straight line, and then we can simplify this part into a easier or a smooth path. But up until this point, we can no longer stretch into a straight line because this path cannot cross. And that would be a definition of a 3-1 knot. And here is another example of a phi 2 knot. So you can see that these helices are simplified into straight lines, but then up until this point, the ends are fixed, but the path cannot be further simplified, and this is then defined as a phi 2 knot. So very different from what you may have learned in the previous lectures in this winter school. What we are trying to investigate are literally or the physical knots formed by linear polypeptide chains. So can we then experimentally prove that these knots do exist? The answer is yes. So for example, you can take a 4-1 knotted protein. You can immobilize one end to the surface here, and then you can use atomic force microscopy scantipeter tips to pull the other ends all the way up. So if a polypeptide chain is unfolded due to this mechanical pulling up until a point that it cannot be stretched into a straight line because of its knot, you will have a shorter contour length compared to the predicted contour length for a linear chain. And that's exactly what this study have found. For this protein, if it's a unknotted, a linear chain, the predicted contour length is about 104 nanometer, but experimentally they observe only 98 nanometer. And that corresponds to a difference of about 17 to 20 amino acid based on force pulling molecular dynamics simulation shown here. So this is a MD simulation result that is a knotted structure that cannot be disentangled. And this compact structure actually corresponds to about 17 amino acids or so. So that's a direct experimental evidence to show people that these protein knots do exist in nature. So these days, there is a server that is widely used in the community called the knotprot, and they systematically update the protein knot entries in the PDB. So earlier last year, there are about 2,000 knotted protein structures reported in the database. That actually corresponds to about 1% of the total protein structures reported in the literature. It's not a huge number, but it's a significant number. It means that in nature, there is a significant portion of the protein topology that are knotted. And they are different types of knots, from 3-1 knot, 4-1 knot, 5-2 knot, up to 6-1 knot. And what do these protein knots do? If we look at some of the large knotted protein families, some of these knotted elements are used to bind to cofactors. For example, YIPK and YPEA. They have pretty different structures if you just look at them by eye, but they have the conserved knotted elements that I highlighted in red and blue. And these knotted elements are used to bind to a cofactor that becomes a methyl donor to modify RNAs. And that they are very important for protein translation because tRNA and ribosomal RNA need to be modified by these enzymes to become active. For the Phi2 knotted proteins in humans, these are ubiquitin C-terminal hydrolases. And the knotted region I color mapped on the surface in blue, they are used to bind to the target protein ubiquitin highlighted in green. There are some curious examples in nature that within the big family, some of the family members are knotted and the other family members are not knotted, as I highlighted in the bottom. So there is AOTCase, it is knotted, this red helix thread through this blue loop, but the majority of these family members are not knotted. So you will see there is a shorter loop shown in green, and this magenta loop and helix are not threading through this loop. Both enzymes are functional, but they recognize slightly different substrates for their enzyme activity, meaning that during evolution, something must have happened so that the topology diverge, or you have a separation of knotted versus unknotted enzymes that recognize similar but not identical substrates for enzyme activities. So I mentioned about AI-assisted protein prediction, AlphaFold. So if you look at the trend of protein structures developed or deposited in the database, highlighted in green, I say NR30, that's non-redundant 30, meaning that if two sequences are very similar, you can basically assume, or you can have high confidence to predict a structure of an unknown protein based on the sequence similarity. But there is a threshold in the field of structural biology saying that if the sequence similarity is below 30%, then you will have more difficulty, or the confidence of the prediction will be lower. So you can see that after the structural genomics initiative launched in early 2000 and finished in before 2010, the conformational space, or the sequence space, is pretty much covered by all the experimentalists. But then the number of structures are still limited. We have in the database about 150,000, less than 200,000 structures. But AlphaFold releases 200 million predicted protein structures. That is 1,000 times more than what we know experimentally in the database. So this huge prediction database potentially contains some new structures, new fold, or new topology that have not been identified experimentally. So a number of scientists then systematically investigated the AlphaFold predictions, and indeed they identified some new protein knot families that are highlighted in the bottom. You have, on the one hand, the so-called composite protein knots. That's the combination of two trephine knots linked together in a single polypeptide chain, and they fold into a compact structure, but then you have two knots existing along a single polypeptide chain. On the other hand, you have two new types of knot. That's the 5-1 knot, and the other is the 7-1 knot. And they have not been identified experimentally in the literature before. And in mathematical terms, these are torus knots compared to the twist knots shown above. So they may have slightly different folding properties compared to those twist knots. So in the last 20 minutes or so, I will give you an overview of how we use experimental strategies to probe protein knotting and folding, and ask the question of why they knot and what if they don't knot. So we talk about the free energy concept of protein folding, and free energy has two terms. One is enthalpy, the other is entropy. So we can broadly define the enthalpic terms as those that are defined by the structure, and then entropy should correspond to the dynamics or the flexibility of the system. The idea, so these days we can carry out molecular dynamics simulation, or basically in silico simulations, to calculate all the energies within the system. So the idea is that if we can define all the energies corresponding to all the conformations within the protein system, we will be able to define the free energy landscape of this particular system. It sounds easy, but for large protein, that is still a very challenging task. But this has been demonstrated for a relatively small protein, and what you will see in animation later is that on the left, that's the conformation in the simulation. On the right, you have a black dot. That's the energy corresponding to that conformation. So as we proceed with MD simulation to find a stable conformation, the black dot should then go down along the free energy landscape that you will see now. So what you will see is that it is initially sampling different high energy conformation, but then it will go into the funnel, perhaps, and then very quickly find a solution that is properly folded with well-defined secondary structure. So this can be done in silico to have a comprehensive atomic insight about how protein folds. But experimentally, it's not so easy to define the positions of individual atoms, especially in a time-dependent manner. There are some tools around, but in general, this is still a very challenging task. So we, as experimentalists, we will probe protein folding events using different readouts or different probes. And that is similar to the idea of trying to describe an elephant by a group of blind men. And depending on where they touch, they may see different things. So it is important to have what we call a multiplex approach using different structural probes to be able to get a better and more comprehensive description about the protein processes of our target protein. So the first tool we use is a far UV circular dichroism, and that basically probes the backbone chirality of protein structures, and that's particularly sensitive to secondary structure, especially helices. So for many other non-biosystems, CD is also very, very useful to define the chirality. The other way we can look at the chromophores chirality, looking at the tryptophan, tyrosine, phenylalanine side chains in longer wavelength range, and that is very sensitive to the tertiary structure. So basically how the protein fold in space and how these aromatic residues are packed against each other or against the other protein structural elements. We can also use the intrinsic fluorescence spectroscopy, and the exposures of these chromophores would change the the spectral signatures. So we can compare the natively folded protein and unfolded proteins. They would have different fluorescence spectra. And by comparing different spectra as a function of time or temperature or a chemical denaturant, then we can define the degree of structure reflected by the intrinsic fluorescence. Or we can use extrinsic fluorescence probes. Some dyes are very sensitive to hydrophobic exposure. For example, ANS would bind to partially unfolded hydrophobic patches. So if a protein is partially unfolded, then the ANS would bind and show a strong fluorescence signal through this binding event. So here is one example. We have a knotty protein, BAP1. It is important in many cancer diseases. There are many naturally observed mutations. So we can see that some of the mutations would change the fluorescence property or the near UV CT spectra. But in general, the far UV signal are comparable, meaning that the secondary structures are preserved, but then the compactness of the protein may differ quite significantly. And that has some functional ramification in terms of the biological readouts. The other tool we use quite a lot is small angle X-ray scattering. And we are very fortunate to have a very good SKCM inline here in Taiwan. And SKCM is very versatile in terms of experimental condition. So we can use the scattering profile to define the shape, the fold, and in some special cases, we can even compare the atomic structures if we have a model available for that kind of comparison. But typically what we use for SKCM is to use the radius of gyration derived from SKCM profile in the low Q region to compare the compactness of our target protein under different experimental conditions. So here is an example. We looked at a series of topologically knotty protein in their native condition, shown in green dots here. So you can see that the radius of gyration of these natively folded knotty proteins show a similar distribution compared to all the other native proteins that are not topologically knotted. We can also denature the protein by chemicals, and then you will see the expansion of the global dimension of these knotty proteins. And curiously, these knotty proteins also show a similar trend following the random coil behavior as predicted by polymer theory. What is interesting in this particular study is that Sophie Jackson in Cambridge, who I collaborated when I was still a postdoc there, she demonstrated for two knotty proteins, you could denature the protein by chemical, introduce two thiol groups to cross-link them under denaturing condition, and remove the denaturant to refold it. This is similar to this Christian Effinson experiment I showed you earlier. If this protein becomes unknotted under denaturing condition, so this becomes a linear chain or it becomes trivial, the cross-linking will then prevent the knotting process from happening when it returns to the native condition. What Sophie found, surprisingly, was that all the proteins are able to return to the native state when they are cyclized under denaturing condition, meaning that these proteins remain knotted or loosely knotted under denaturing condition in their experimental setup. We tested the same sample under the same condition and investigated them by small angle X-ray scattering. We found no preferential compaction of this knotty protein compared to other unknotted protein, meaning that you could have a loosely knotted polypeptide chain under denaturing condition. They behave like random coils, but this knot would not disentangle for some reason. So exactly what is keeping this knot closed under this denaturing condition is still something to be explored. We still don't know the answer to that. But this is a clear indication that this knotted protein remain knotted under denaturing condition. So I will not go into the details of what we did. These are published, but I just wanted to show you that to characterize protein folding pathways, we routinely combine different tools: intrinsic fluorescence, far UV-CD, light scattering to look at oligomeric state. In here, we look at the smallest knotted protein known in the literature when we started the experiments. It unfolds into a native-like folding intermediate as a monomer and then becomes fully unfolded. In the other extreme, we have a 6-1 knotted protein. It's pretty large, more than 300 amino acids. We combine far UV-CD, intrinsic fluorescence, small angle X-ray scattering. And so to analyze them globally, we discovered that this protein will have 2 folding intermediates that have different spectral signatures or SAC signatures, and then eventually becomes fully unfolded in the denatured state. In the other example, we can even map out the sort of the reaction coordinate as a function of the structural RMSD, and our experimental finding actually matches very well with the simulation results that are published before us. So the take-home message is that all the protein or all the knotted protein that we investigated so far, they all exhibit highly populated folding intermediate, which I indicated as I. Some of them have parallel folding pathway. Some of them have sequential folding pathway with distinct folding intermediates. Some are more complex with bifurcated folding pathways from the native to the denatured state. Folding intermediates are actually problematic for proteins because you can imagine if you start from an unfolded high energy conformation going to the native state, you're sort of trapped in the local minima, but you could go off pathway into a misfolded conformation. And that's the result, or that's the cause of many of the neurodegenerative diseases, such as Parkinson's disease and Alzheimer's diseases. Now, in one particular family, we look at the 5-2 knotted human ubiquitin C aminohydrolases. In humans, we have 4 paralogs. We compare the folding kinetics of these 4 different proteins. What we found is that there is one particular protein called UCHO5. It has very, very slow intrinsic unfolding rate. Under native condition, it only unfolds once every 100 million seconds. Basically, within the lifetime, it probably does not unfold, or very, very rare. This protein is an enzyme to remove ubiquitin from the proteasome. And proteasome is a large machine to digest misfolded protein. So you can imagine this is like putting your hands in the meat grinder. You have to be very careful to catch the substrate before you are actually pulled into this proteolytic chamber. So we speculate that this knotted UCHO5 should be mechanically very stable to withstand the force pulling generated by these proteasomes. So we set up, so this is the way we imagine how it works. So you have a polypeptide chain, you are almost getting pulled into the proteolytic chamber. If you tie a knot, then you are sort of more stable mechanically to withstand the force pulling. So the experiment we designed is we introduce a small tag called SSRA tag. This tag is recognized by a bacterial proteasome called ClipXP. You can monitor the reduction of the substrate intensity as a function of time to evaluate how stable is this protein when it is subject to force pulling coupled degradation. So in the field, green fluorescence protein is a gold standard. It's a very, very stable protein, but it will be degraded within 20 to 30 minutes. In contrast, for our Phi2 knotted UCHs, after 3 hours, we don't see much reduction, suggesting that this protein is mechanically very stable compared to green fluorescence protein. However, green fluorescence protein has very high thermal stability. It is also chemically very, very stable, whereas these Phi2 knotted proteins, they will unfold at around 50 degrees or so, meaning that for these proteins, their chemical stability or thermal stabilities do not correlate with the mechanical stability generated by their topological constraints. What is also surprising is that these proteasomes or these proteases use ATP to generate energy to pull the substrate. Once we introduce UCHO5 to the system, the ATP turnover rate dropped dramatically. So it basically jams this molecular machine in such a way that it is no longer to hydrolyze ATP to generate energy to unfold the substrate and degrade it, which is actually very, very surprising. So the other aspect of knotted protein study that we are doing, and this is actually sponsored by SKCM² for Qi Jun's postdoc fellowship, we are asking the question of what is the evolution or evolutionary trail of these knotted proteins. And the system we look at is a family of RNA methyltransferases called the SPOUT family that I showed you before. We have YBEA and the YPK. They look quite different, but they share the same motif to bind to this cofactor as a methyl donor. In 2007, there was a bioinformatics study. They predict that the last universal common ancestor, known as LUCA, so at the beginning of evolution, this protein is also knotted based on sequence comparison. So why would nature develop such a complicated protein topology for their functions in the early days? So coming back to the definition of protein topology, which is basically the sequential order of secondary structures. As I highlighted here, you start from the red beta strand, ended up in the purple beta strand. We can make a circular permutant by protein engineering. So we close the original N and C termini and introduce a new opening. So in the context of protein topology, this protein will then, or the circular permutant will then have a different topology. What I will show you later is that we can even cyclize the protein. And that introduces a puzzle or a conundrum in protein topology. That is, if you don't have an origin, how do you define protein topology of this cyclized protein? So the two questions we ask is, can we untie a protein knot by circular permutation? And the second question is, what will happen if we make a true mathematical knot? Because proteins in general are not closed paths. So mathematically, they are not, strictly speaking, a real knot. So what we did was we used protein engineering. We introduced new opening in different places and see which one can fold. And in the first case, we could have a circular permutant that has, by eye, the same native structure compared to the parent knotty protein. But what happens is that we introduce a new opening, and then this protein, by the definition of protein knots, is no longer knotted. We can look at the cofactor binding pocket. In fact, if you look at the side chain orientations of the parent and the circular permutants, they look very similar. But because of the opening, this circular permutant can no longer bind to the cofactor. In the other extreme case, once we have a circular permutant, one half of this molecule flip to the other side to become a domain-swapped dimer. And again, this protein can fold, but it can no longer bind to the cofactor. So we believe that the knotted element is essential and functionally important to bind to its cofactor. And this is the job that it was designed to do in the early days of the evolution. Now, we can also cyclize the protein by enzymes. So we have a protein purified and we close the N and C termini by an enzyme called sortase A and solve the crystal structure, and it looks identical to the open chain protein. But this cyclized protein then now has a thermostability increase by more than 20 degrees Celsius when we unfold the protein. And this protein can bind to its cofactor in exactly the same way as the linear chain. So the cyclization basically highly stabilizes the linear polypeptide chain without perturbing its biological function. What is interesting is that if we use small angle X-ray scattering to compare the radius of gyration for the linear and the cyclized protein, which we derive by the linear approximation, what is interesting is that we look back in the old literature. Kramers has a polymer model to predict that the radius of gyration, the ratio of the cyclized form over the linear form, the squared, should be 1/2. And that's exactly what we got in this experiment. It is important to note that for organic chemists to synthesize homogeneous lengths of cyclized polymer, it's not always very easy. And usually this polymer analysis are done for homopolymers. But proteins are heteropolymers with 20 different kinds of building blocks. But in these experiments, we show that despite the fact that the side chains are heterogeneous, the unfolding process or the random coil behavior of these heteropolymers follow exactly the same behavior as Kramers predicted for cyclized polymers. We did recently, the paper was accepted just last week. We repeated the same for another system, and then the same result was found, indicating that this kind of trend or this polymer characteristics is applicable to very heterogeneous heteropolymers, such as a protein of different sequence composition. And we can use that as a model system to look at some of the polymer physical properties. So to summarize, for this knotty protein, we investigated their mechanical stability. We showed that we can untie the protein knots by circular permutation, but then the function is lost. We can also cyclize them to make them as true knots, And then we observe some interesting physical properties of these true mathematical protein knots. I don't have the time to talk about the simulation we did together with the Italian group, but then we for the first time, have a converged view from experiment and theoretical calculation about how protein may knot in the transition state. So finally, sorry about the overtime, but in the last couple of minutes, I want to tell you that because of AlphaFold, scientists have identified new protein knots, such as this 7-1 knot or the 5-1 knot. The question is, are they real? Can we trust the AlphaFold predictions? So we carry out a series of structural analysis for the most complex 7-1 knot by X-ray crystallography and NMR spectroscopy. Unfortunately, in a way for us, AlphaFold prediction is correct. The experimental structures determined by different approaches are essentially identical to the AlphaFold prediction, with the exception that the N-terminal flexible region is not well defined. And AlphaFold also says that it has low confidence. But you will see this 7-1 protein knot is very compact. So there's not a lot of space for it to move around. And we use the same mechanical unfolding analysis, and this 7-1 protein knot has comparable mechanostability compared to that of UCHO5, which was the record holder that we discovered some years ago. In the literature, theoreticians try to simulate the pooling process or the translocation process of polymers through a nanopore. What they found was that of all these different knots, the torus knots are more stable than twist knots. And indeed, the 7-1 protein knot that I show you just now belongs to the torus knot. Thanks to Yvonne, who pointed out the paper from Decanes about the tight knots. We looked into the structural features of this 7-1 knot. It is actually probably a model system to look at these tight knots as shown on the right by the original paper here. The idea is that you can pull this linear chain into a tight structure that you cannot move around. It may slide, but it is not loose. And there are some intriguing features of these tight knots that can be investigated. The experimental evidence of tight knot was reported that I showed you earlier by AFM pooling experiments of a 4-1 knot. And in their case, the tight knot size is about 17 to 20 amino acids. If we look at the literature, the tight knots for 3-1 knot, 4-1 knot, 5-2 knots, they have the size that I color here in blue, relatively small. But the crystal structure or the experimentally determined protein structures that are known in the literature, they are pretty large, suggesting that in addition to the regions that are the minimal building blocks to form such kind of topological knots, there are additional secondary structures or loops that are present, and that may give some more space for the protein to tie a knot. But the last example that I just showed you, the 7-1 knot, if we follow this trend, it is really tight. There's not much space for this protein to move around before it falls into the native structure. So we are interested in knowing how can such a small, complex, and stable knot be formed spontaneously in nature. So we have some outstanding questions. One is there a conserved promoting signature? And that's the question that will be addressed by Qijun hopefully in the near future. and what is the evolutionary trail of different protein knots. And what probably most exciting for us is, can we design new protein knots with novel functions that may help us improve the quality of our lives? So with that, I would like to thank all my team members, Yvonne and Professor Tate, for the opportunity to join the SKCM² and sponsor Qijun's postdoc fellowship, and all the collaborators and staff within Academia Sinica to support this work. Thank you.